Good afternoon. Welcome back to Virology. Today we're going to explore the relationship between viruses and cancer. This is called transformation and oncogenesis. So let's do an experiment first to define transformation. So we take a hamster embryo, we cut it up and treat it with trypsin so we get single cells, and then we plate those in plastic dishes and cover them with medium so that they will grow. And if we do nothing to the cells, they will grow normally, and they will look like this first panel on the right. That's a microscope view of what the cells look like. They're normal. Normal means a specific thing, which we'll get to in a moment. But if we take the second plate of cells and treat them with a mutagen, like a mutagenic chemical, then some of the cells become transformed. And they look like this on the right here. And they're very easy to see. They grow very large, and they grow on top of one another, and they're generally disordered, as you can see, compared to normal cells. One of the things that normal cells do is when they touch each other, they stop growing, but the transformed cells keep growing. So we can see transformed cells in the laboratory very easily. Now, in cells in culture, this is another view of transformation. On the top, we take mouse cells. So we do the same experiment with the mouse embryo. We make single cells and we plate them out in culture. And we're looking here at the cell growth rate versus the day after you start the culture. And you can see the mouse cells, they will grow, but then they start to die, slow down in growth. They go through a period called crisis, and then they emerge from crisis and begin to grow again. And these cells that grow are transformed. We didn't treat them in any particular way, but mutations arose spontaneously that made the cells transformed. Now, if you do the same thing with human cells, you could take human foreskin fibroblasts, for example, and you plate them out. They will grow for some generations, and then they will die. You never see transformants emerge unless you do something to the cells like we did previously. Now, why is this difference? This difference is illuminating, actually. It has to do with telomeres. Telomeres, of course, are the ends of the chromosomes. And here's the end of a chromosome, and the telomere is protecting it. In fact, the telomere is a complicated structure consisting of a variety of proteins, including telomerase, which contains a reverse transcriptase activity. During embryonic development, this telomerase is active in all cells, and the ends of your chromosomes get longer and longer until you're born, and then telomerase activity stops. And you're born with the length of chromosome ends that you're going to have the rest of your life. And most mammalian somatic cells do not produce telomerase. So that's why human cells die eventually in culture. With each cell division, the ends of the chromosome get shorter and shorter because of the end problem in DNA replication. The end gets shorter and shorter, and when it gets to about 4 kb, the cells die. That's why those human cells died in culture. Mouse telomeres are longer than human telomeres, so they can grow more generations. That increases the chance of random spontaneous mutations leading to immortality. And one of the ways you could become immortal is by making telomerase again. So you could have a mutation that reactivates telomerase, and now the ends get longer, so the cells just keep dividing forever. And we can do that in the lab. We can take the telomerase gene and put it in cells and make them immortal. So if you want to be immortal, take some of your cheek cells, put them in culture, put telomerase in, they will grow forever, okay? They will outlive you if you need that. I know someone who probably would like that. He's got orange hair. Okay, so that's what's happening at the ends, and that's why cells die in culture. So here's, and telomeres have to do with cancer also. And so, you know, in regular cells, as I've just told you, here are the telomeres, the ends of the chromosomes. With each cell division, the ends get shorter and shorter, and eventually somatic cells stop growing. But cancer cells grow all the time because the telomere doesn't get shorter because there are mutations that have allowed the telomere to remain the same. That's one of the kinds of things that can happen to cause cancer cells, having the telomere active all the time. All right, back to transformation. So a transformed cell has a number of properties, including it's immortal. It will grow indefinitely. And HeLa cells, the famous HeLa cells that are in many labs, which were taken from Henrietta Lacks, as we'll see later, are immortal. They have loss of anchorage dependence. They don't have to grow stuck on a plastic dish. Most cells like to grow on a plastic dish, or at least attached. Transformed cells don't care. They lose contact inhibition, which means normal cells, when they touch each other, stop dividing. Transformed cells pile up. You can see here on this A and B. A, the normal cells are touching each other, and B, they're piling up on one another. Transformed cells can also make colonies in semi-solid media like agar. You can suspend the cells and they will grow, but normal cells can't grow on their own. They can't handle it. The growth factor requirements are too great. And finally, transformed cells have decreased requirements for growth factors. Now, the reason you put growth factors, the reason you put serum on cells in culture, the medium has like 5 to 10% serum, because serum is rich in growth factors. And normal cells need that to divide. But transformed cells have reduced requirements for growth factors. All right, so those are the differences between normal and transformed cells. Now, remember, the title of this lecture is Transformation and Oncogenesis. What's oncogenesis? They are two different things. Oncogenesis is development of cancer. And a tumor is a mass or a swelling produced by abnormal growth of cells. They keep growing and growing and growing, and they make a mass. And that mass can stay in place, or cells can break off and go throughout your body, in which case it's malignant. Cancer is a genetic disease. You will see aspects of that today. About 8 million deaths per year in developed countries. We don't know the actual cancer rate because healthcare systems in many countries are not good enough to report all the cancers that are occurring. And I said it's a genetic disease. You need about 12 mutations in a variety of genes that control cell division, cell survival, maintenance of genome integrity, which means error correction. You need about 12 mutations, and a cell will become a cancer cell. Now, you may be born with 11 mutations that you get from your parents, and during your lifetime, you get one more. How can you get one more? Environmental carcinogens, big one. Air pollution, radon, smoking. They can make that last mutation and you get 12. Or you can be born with none. And then maybe you will live your whole life without cancer. You know, 1 in 3 people get cancer in our lifetimes. So the mutations, besides inherited, they can be caused by mutation, DNA damage, UV light, for example. And, you know, the mutations, DNA synthesis is mutagenic, as you know, so that's part of it. Or viruses. Viruses can contribute to those extra mutations. Whether you need 12 or 6 or 1, viruses can help do that. Okay, so that is the difference between transformation and oncogenesis. They are distinct. Transformation just says a cell line has become immortal. But sometimes if you take that transformed cell, so on the right, and inject it into a nude mouse, you have to use a mouse without an immune system, because most of the time when you put cells in a mouse, it will reject them. But if you put it in a nude mouse, sometimes the transformed cells will cause a cancer. That needs, as I've just said, it needs other genetic changes in addition to transformation, about a dozen, for that to happen. Today, we're going to talk about how, by studying virus-transformed cells. So yes, viruses can transform cells, just like mutagenesis can. Studying viruses and transformation of cells by viruses explained to us exactly how cancers arise. We really had no clue before people figured it out with viruses. And now all kinds of cancers make sense, because not every cancer is caused by a virus, right? But studying virus-caused cancers provided incredible insight, and that's what I'm going to try and show you today. But the virus actually doesn't cause cancer itself. Infection leads to cancer, or can lead to cancer. A virus can transform a cell, and then the rest is up to genetics, right? Usually when a virus transforms a cell, what happens is the cell divides forever. And with each division, mutations are accumulated. And then, if those 12 mutations are fulfilled, then you have a cancer. So viruses transform cells, the rest is up to the cell. So viruses are contributing factors in about 20% of human cancers. And these are the human cancer viruses. There are both RNA viruses and DNA viruses. And you can see the names of the viruses here and the kind of cancers that they can cause. So HTLV, HIV, hepatitis C virus are the RNA viruses, Epstein-Barr, Kaposi's, sarcoma, herpes virus, hepatitis B virus, human papillomavirus, and Merkel cell polyomavirus. These are the viruses associated with human cancers. I'm not going to talk about any of these today, except for 5 minutes, human papillomavirus. But what I'm going to talk about is how we came to understand how they transform cells. By studying other viruses, and that is viruses that cause tumors in non-human animals. Now, over the years teaching this course, I used to say transformation and oncogenesis caused by virus infection is not required for the replication of any virus. It's an accident, as you'll see by the end of this lecture. And then one year, someone emailed me who works on a retrovirus that causes tumors in fish. This fish is a walleye, and the virus is walleye dermal sarcoma virus. And that's the only example we know where making a tumor is actually needed for virus transmission. I'll talk about that later. So now I have an asterisk by Annie because of that nice person who emailed me. So today's story begins in 1909. A farmer from New Jersey brings a hen to Dr. Peyton Rouse. He's working at the Rockefeller Medical Institute. That's what it was called before it became Rockefeller University. And this chicken has a tumor. It has a solid tumor, which he was trying to figure out what caused it. And he thought it might be a virus. So he cuts out the tumor. He grinds it up. He puts it through a filter. Remember, this is 1909, so we now know that viruses can go through filters. He takes what goes through the filter, he injects it into another chicken, and that chicken develops a solid tumor. So he showed that viruses could cause cancer, although he wasn't the first, because others had done it before, but we didn't really understand it. It took 50 years for him to get a Nobel Prize for this. Nobody believed it at first. People thought other things caused cancer, but eventually others worked on this and they provided the evidence showing that yes, in fact, his virus causes cancers in chickens. He got the Nobel Prize in 1966. His virus that he discovered was called Rouse sarcoma virus because the chicken developed a sarcoma, and of course his name was Rouse, or RSV. It played a role in 2 more Nobel Prizes, as we will see today. Okay, so this whole story actually is very well told in this book, The Emperor of All Maladies by Siddhartha Mukherjee. I'm sure some of you have read this. He tells this story about cancer discovery. By the 1950s, cancer researchers have split into 3 fueling camps. The virologists, led by Rouse, claimed that viruses cause cancer, though no virus had been found in humans. Epidemiologists argued that chemicals caused cancer, but they didn't know how. And the third camp said, we have genes in us that actually caused the cancer. All 3 were right, as you will see. But it took Howard Kemen to start us on the road to understanding that. We've encountered Howard Kemen before. He co-discovered reverse transcriptase. In 1950, when he went to Caltech to work with Renato Del Becco, who developed a plaque assay for eukaryotic viruses, he was going to study fruit flies, but he got bored with it, and he decided to study Rouse sarcoma virus. Why? So in Del Becco's lab, he realized that you needed to study virus infections in cells in culture, not animals. That wasn't going to tell you a lot. And cell culture had just been developed in the early 50s, and Del Becco was an opportunist. He said, we can use this to study viruses. And then Kemen said, well, we could use chicken cells to study Rouse sarcoma virus. And so that virus had only been shown to cause tumors in chickens, but he thought he could do something in cells in culture. And after 7 years of doing this, he worked it out. He succeeded. And back to Mukherjee, this on the top, he added Rouse sarcoma virus to normal cells. Infection incited them to grow uncontrollably to form tiny distorted heaps containing hundreds of cells that he called foci. Foci, Kemen reasoned, represented cancer distilled into its essential elemental form, cells growing uncontrollably, unstoppably, pathological mitosis. So that's it. That's transformation, right? He showed that the virus, when added to chicken cells in culture, could transform them, and that gives them the properties I just told you. Here are some foci of transformed cells, because the whole monolayer isn't transformed. You get a focus where a virus landed and infected the cells. These are fusiform, these are refractal and round. These just look different. And here's how the foci look. The normal cells down here, and then wherever a virus infected a cell, it made a pile of transformed cells. So these are the avian cells transformed by Rouse. This is a revolution in our ability to study cancer. Now, this wasn't the only discovery that is important for this whole story. DNA viruses were also found to cause transformation. So Rouse sarcoma virus, we now know is a retrovirus. It's an RNA virus. But polyomavirus and SV40 were shown in the 60s to cause transformation of cells in culture. Most of the cells died, but rare cells did not, and they grew out into colonies, and they were transformed. So both RNA and DNA viruses now are shown to cause transformation of cells. How can a virus transform a cell? There are 3 requirements. First, you can't kill the cell, right? The virus kills the cell. How can it live forever? So cytopathic effects have to be eliminated. Second, you have to reduce virus particle production, which can also kill cells. And in fact, transformed cells do not produce virus particles. And third, the cell has to continue to divide. It becomes immortal. Does this ring a bell? It's a certain kind of persistent infection, right? The genome goes in, stop producing virus particles, yet something's happened to the cells to make them live forever. And we have RNA and DNA viruses, and we are going to talk about those separately and how it's working. But the first question is, which of the following is not a property of transformed cells? Increased requirements for growth factors, immortality, loss of anchorage dependence, loss of contact inhibition, colony formation in semi-solid media, which is not a property of transformed cells. All right, how did we do? Right, number one is the correct answer. Increased requirements for growth factors, not a property. It's decreased requirements for growth factors. Immortality is clearly a property, and colony formation in semi-solid media is also a property, as I've told you. So here's the overview of what we're going to talk about today. Our understanding of viral transformation of cells in culture and how that relates to cancer formation. And we're going to do it with RNA and DNA viruses. We're going to use retroviruses as an example. That was begun by Rouse. We're going to talk about DNA viruses, the polyomas in SV40s causing transformation of cells, and also people studying in vitro cancer biology. These are going to converge in the 60s and 70s. And today, our understanding of cell growth control is totally because of these viral studies. Completely. The whole way that cell division is controlled, we understand because of studies of viruses. It's really quite remarkable. Okay, how does Rouse sarcoma virus cause tumors in chickens and transform cells, right? Two independent things. Let's tackle that first. First, what you need to know is that in 1908, two scientists, Ellerman and Bang, had shown that avian leukosis virus causes leukemia in chickens. Right, avian leukosis virus, white blood cell tumor, which involves proliferation, but no solid tumor. So people at the time didn't think it was actually a cancer. We didn't realize till later, yeah, it's a cancer, it's just a different kind of cancer. So they were actually the first to show that a virus could cause cancer. This virus, avian leukosis virus, infects virtually every chicken on the planet. You're worried about H5N1? We don't actually have to worry about this, but it is in every chicken. And most of the chickens get infected after hatching. They get it from other chickens in the flock. And the older chickens, more than 14 weeks of age, about 3% of them develop leukemia or leukosis, another word for leukemia. Almost all the birds get transient viremia after infection, but most do not get leukemia, only a few. And you'll see why in a bit. Okay, that is avian leukosis virus. Birds, if you let birds live, most of the time they're killed at a certain age. But if you let them live, they'll develop solid tumors like sarcomas. And if you take the virus from these tumors like Rouse did, they cause tumors. They cause sarcomas, not leukemias. And so Rouse isolated a virus from one of these solid tumors called Rouse sarcoma virus. He was lucky because this was not a defective virus, but most of the isolates you get from solid tumors are defective. What do I mean by that? I have to tell you a few other things before I explain it. So Rouse is actually derived from avian leukosis virus. The chickens are all infected with avian leukosis virus, and some of them go on to cause cancers, leukemias, or if you let the birds get older, solid tumors. So avian leukosis virus is the parent virus of all these leukemias and sarcomas. So Mike Bishop and Harold Varmus at UCSF began to study this question. Why does RSV cause sarcomas but not ALV? So they compared the genomes of the two viruses, and they found that, in fact, the genomes from solid tumors like Rouse sarcoma virus are recombinants. A piece of the viral genome is replaced with a piece of host DNA. they called it an oncogene, and that, they said, gives Rous sarcoma virus the property to cause solid tumors. They got the Nobel Prize in 1989 for that discovery. The other Nobel Prize is to Howard Temin, who discovered reverse transcriptase in Rous sarcoma virus. So now, Harold Varmus, by the way, was head of NIH for 10 years under Clinton. He's now at Sloan Kettering, working on sarcomas, of all things. So ALS, avian leukosis virus, picks up which causes leukemia, picks up a piece of host DNA, and now it causes sarcomas. So what's this piece of host DNA? All right, so remember, ALV-infected birds come down with a variety of tumors if you let them live. Initially, they get leukemia, and then if you let them live longer, the others develop different kinds of tumors. They all contain viruses derived from ALV, and they're all different, and most of them are defective, which means that the host gene has replaced one of the viral genes, so it can't reproduce on its own. It needs a helper virus. So Rous was a lucky guy. His virus grew on its own. It was not defective. It had all the viral genes. Now, furthermore, if you look at different tumors from different chickens, they all have different pieces of host DNA. Now, in RSV, Bishop and Varmus called the host gene SRC, SRC, for sarcoma. But all the other genes were different. So they're called oncogenes in the host, right? Which is not a good name because they're not there to cause cancer, but they do. So they called them oncogenes. And over the years, many people identified different oncogenes from chickens or other animals that develop tumors when infected with retroviruses. It's a complete gold mine for molecular oncology. And here are some of those viruses. So on the left, we have avian leukosis virus. That's the virus that infects every chicken in the world and causes some leukemias. And if you let the chickens live, they develop other tumors like sarcomas, which Rous worked on, myeloblastomas, myelocytomas, erythroblastomas, reticuloendothelia, all different kinds of cancers. And if you do the Rous experiment, you find a different cell oncogene in each virus. So here's Rous sarcoma. There's the avian leukosis virus genome. GAG, POL, 3 genes. Rous has SARC stuck at the end, so it's not defective. But look, this one, MIB, the cell oncogene MIB is stuck in envelope, so this virus needs a helper to make particles. You can see different oncogenes stuck into different places. They're all defective. Yet, all of this identified to us cell genes involved in growth control. Amazing. On the right are mammalian transducing viruses. You could do the same experiment in mice or cats or even non-human primates. So you can see murine leukemia virus, GAG, POL, envelope. You can isolate different kinds of cancer-causing viruses, leukemia, sarcomas, in mice and in cats. And you can isolate viruses from them that have picked up a cellular oncogene, which makes them cause cancer, which makes them transform cells and eventually leading to cancer. So over the next 20 years, this is what people did, and they identified all of these genes that regulate cell growth. Now, just a few words on defective versus non-defective, right? Defective viruses need a helper, another virus to help grow, because the cellular oncogene has gone into one of their genes and disrupted it. Usually they're missing envelopes, and these genes are deleted during oncogene capture, which I'll show you in a minute. And there's an example of avian leukosis virus. Rous sarcoma, SARC went in after envelopes, so it's completely non-defective. But for avian myeloblastosis virus, the MIB oncogene, and these are named according to the cancers, by the way, right? SARC, MIB is myeloblastoma. MYB. It goes into the envelope gene, so this virus can't make particles without a helper. That's what defective means, okay? Here's a slide on how you would pick up oncogenes, okay? Remember, retroviruses infect cells. They make DNA, which then integrates into the cellular genome. And by chance, of course, they're going to integrate next to one of these oncogenes, like SARC or MIB, right? And by chance, transcription is going to, at some point, include the oncogene in the mRNA. It'll be built into the virus particle and eventually make its way into new cells. It'll be delivered to cells by virus particles. So it's an accident of the fact that retroviruses have to integrate their DNA into the host cell. There's no need to transform cells. It's an accident. If integration is random, eventually you're going to hit an oncogene. So the genes in the cells are called oncogenes, actually proto-oncogenes, because in the cell they control cell growth. They don't cause cancer. They're highly regulated, and we give them names like CSARC for cellular SARC. And retroviruses will pick these up, and in some cases the oncogenes will become mutated, and that makes them even more able to cause uncontrolled cell growth, and we call them like VSARC. And we've got about 60 so far of these. And if you remember the cell cycle, the 24-hour cell cycle, there's mitosis there in red. We have a period of cell growth and DNA synthesis, and then mitosis. Proto-oncogenes control whether a cell will divide or not. They give the signal to the cell. That's one of the several signals that we're going to see today. So that's why they're highly regulated, because if they're around, if they're expressed, the cell's going to keep dividing, and that's not good. So here's what the oncogenes do. How do you regulate cell growth? We know this from studies on viruses, but let's look at that first. So on the cell surface, there are receptors. They're called growth factor receptors, and they are receptors for growth factors. So when you have a meal, the food you eat has growth factors in it. It will diffuse to your cells, bind these, and your cells will divide. Then the way the cells divide is that the binding of the growth factor to the receptor causes a signal transduction pathway. So there are receptor tyrosine kinases, there are non-receptor kinases, there are serine threonine kinases. They transduce a signal into the nucleus, and even the transcriptional regulators are encoded by these oncogenes. And this turns on cell growth. So growth factor binding turns on transcription of genes involved in cell growth. If you look on the left here, you can see all the oncogenes that have been identified. And some of these are receptor protein tyrosine kinases. Some of them are non-receptor tyrosine kinases, and that's exactly what SARC is. Some of them are transcriptional regulators. So they all have a specific function in regulating cell division. And if they're overproduced by a retrovirus, that makes the cells grow forever. They're transformed, and maybe they'll lead to a cancer. Now, among these oncogenes, many of these are mutated in human cancers. And so you can imagine if there's a mutation in a growth factor receptor, the cell will divide without the growth factor. It keeps dividing. That one mutation, one amino acid change, makes the cell divide forever. It's called the cancer driver. And these are present in many human cancers. So that's what we have learned from studying how viruses transform cells. All right, so that's the cell cycle again. These proto-oncogenes are involved at telling the cell whether to go into mitosis or not. And we discovered those by studying transforming retroviruses. That we call them dominant oncogenes. You just need one DNA copy in a cell, and that will make the cell continue to divide. So if you put any one of those oncogenes in a cell, it's going to turn the light red, and the cell is going to just keep dividing. These retroviruses transform cells by 3 mechanisms. Now you're going to learn about avian leukosis virus. So the first is rapid tumor formation in about 2 weeks. You inject the chicken, 2 weeks later you got a tumor. That's Rous sarcoma virus. It has an oncogene in its genome. When the virus infects the cell, it produces the protein. The cells start to divide. And remember, retroviruses integrate into the host, so they're there forever. They don't kill the cell. They make it divide forever. So that's the first kind. Intermediate kinetics, a couple of weeks or months. Avian leukosis virus, the parent of RSV, takes a couple of months to cause leukemias in chickens. There's no oncogene in ALV, but it integrates next to an oncogene and turns it on. And that's why it takes a little bit longer. So ALV first causes leukemias, and then chickens that survive and live longer, you get other kinds of solid tumor viruses. And third, very slow kinetics, years. Human T-cell leukemia virus, for example. There's no oncogene in the virus. It does not integrate next to an oncogene, but rather it makes a viral protein that activates oncogenes in trans. And so that's how it makes cells divide forever. And these 3 schemes are shown here. There's Rous, rapidly transducing, it carries an oncogene. ALV, cis-activating, it integrates next to an oncogene. There's no oncogene in the genome. And the slow ones, no oncogene, doesn't integrate next to an oncogene, but it makes this protein X, which is a transcriptional activator needed to turn on transcription of viral genes, but it also has off-target effects. It will turn on cellular genes like IL-2. IL-2 makes T cells divide. They divide forever because the HTLV is integrated, and eventually you get leukemia. So one exception to the fact that transformation is not needed for viral reproduction. Retroviruses transform cells as a mistake. Well, it's not their mistake. They're just integrating randomly, and sometimes they integrate next to an oncogene, right? There's no obvious requirement for transformation or oncogenesis except for walleye dermal sarcoma virus. This virus infects the fish, and they get a tumor. You can see it there. In the fall, this tumor is full of virus. In the fall, the tumor falls off into the waters, and the virus then can infect other fish. So there's an example where making a tumor is needed to transmit the virus to other hosts, but that's only for this one, as far as we know, no others. Fish are fine, by the way. They don't have any problem with this. Well, at least they don't have any signs. They do, I'm sure they have symptoms, but they can't tell us, right? Next question. Which of the following allows Rous sarcoma virus to transform cells? The presence of an envelope, the presence of a pol, the presence of a sarc gene, presence of LTRs, none of the above. Rous sarcoma virus. How did we do? Most of you got the right answer. C, presence of a sarc gene. That's what allows it to transform cells. Not the LTRs. It's not none of the above. It's the oncogene itself that allows it to transform cells. All right, now on to the DNA viruses. How do they transform cells? So the first oncogenic DNA virus discovered was a papillomavirus. You know, human papillomaviruses. They are DNA viruses with a circular double-stranded DNA genome. This is related. It's a papillomavirus that causes warts. So a papilloma is a wart. This causes papillomas in cottontail rabbits. That's discovered in 1933. So these are warts on the rabbit's head and face caused by the virus. Here's another one here. They're fine. The rabbits are fine. The warts fall off eventually and they're okay. It doesn't kill them. But when people see these in the woods, they think it's a mythical creature called the jackalope, a jackrabbit and an antelope. Have you ever heard of a jackalope? No? All right, well, that's in popular culture. Here in New York a couple of years ago, there was a concert called the Feast of the Jackalope, and there's the jackalope, right? Supposedly JC, but it's obviously a antelope, a rabbit. You can see rabbit ears and an antelope with the horns. So this pervades popular culture. Just Google it, you will see. People actually think these exist. They don't. They are just rabbits with papillomas, okay? Everything can be explained by science. So Ludwig Gross, so that was Shope, Richard Shope, who discovered those rabbit papillomaviruses. Ludwig Gross discovered murine polyomaviruses in 1953, about 20 years later. These cause rare tumors under certain conditions. So the natural host for these viruses is the mouse. They don't cause cancer in mice. They infect all mice in the wild, but they don't cause cancer. But they make tumors in other animals that are not the right hosts, like hamsters, rats, and rabbits. They cause tumors of many different tissues, which is why they're called polyoma, many tumors viruses. You'll understand all of this in a moment. Another DNA tumor virus, SV40, which you've heard about a lot, was discovered by Eddy and Hilleman in the 1950s as an early contaminant, actually 1962, an early contaminant of poliovirus vaccines, which we didn't know it was there. We got rid of it right away. And several million Americans were infected with the virus after polio immunization. No evidence that causes any tumors in humans. But this virus will, so the natural host is the monkey, non-human primate. It does not cause tumors in monkeys, does not transform monkey cells in culture, but it will cause tumors in, say, newborn hamsters. So again, we have these viruses that are causing tumors in the wrong host, not the natural host. And that's summarized here. These are responsive cells to infection. So we have the species of cells, we have the virus and SV40 and polyomavirus, mouse polyoma. So the natural host of SV40 is monkeys, so the cells are completely permissive, they die. They're lysed by virus infection, no transformation. Mouse cells are not permissive, so the virus doesn't get anywhere. The mouse is non-permissive for SV40 and completely permissive for mouse polyomavirus, so the cells die, no tumors, no transformation. And these two here with the red asterisks are where we see tumors, and in cell culture, we see transformation. So SV40 is semi-permissive in hamsters. Mouse polyoma is semi-permissive in both hamsters and rat. What does semi-permissive mean? So permissive means the DNA gets in and nothing happens, doesn't do anything. Semi-permissive means you get up to DNA replication and then it stops. So you can make some proteins. That's important. You can make some proteins. And if I'm talking about before DNA replications, those would be early proteins, right? SV40, adenovirus, early proteins. Now, when you infect cells with these viruses, transformation is pretty rare, about 1 in 100,000 infected cells. Why is it rare and how does it relate to tumor formation? We're going to explore that right now. First, though, we have to look at one more family of viruses, the adenoviruses, which do not cause cancer in humans, but certain serotypes are tumorigenic in hamsters. And again, like polyoma and papillomaviruses, transformation in tumors are very rare events. A key finding that people made when studying all of these viruses in transformed cells and tumors is that there's always a viral T antigen present. in tumors and transformed cells. For SV40, that would be T. Remember large T antigen? Polyomas, there's T antigens. Papillomas, they're T antigens encoded by E6 and E7 genes. And adenoviruses, the T antigens are the E1A and E1B proteins. T means tumor, because these proteins are found in all tumors and in all transformed cells. But these T antigens are all different proteins, by the way. They're not the same protein. So how is that working? Well, for all these viruses, T antigens are essential. Remember, for SV40, T antigen has to bind the origin and recruit the DNA synthetic apparatus of the cell. So they're needed for replication. They can activate viral transcription. They're needed for viral DNA synthesis. Of all the viral genes present in a transformed cell or a tumor, T antigens are the only viral genes that are always present. You might have random other viral genes present, but not always. These T antigens are the only ones that are found in every tumor cell, every transformed cell by adenovirus, SV40, polyoma, papillomaviruses. Furthermore, T antigen can transform cultured cells. So remember I told you if you want it to be immortal, take cheek cells and put telomerase gene in them. Well, you can also put T antigen of SV40. People do that all the time, and they will be immortal because T antigen is what transforms the cells, as I'll show you in a moment. So a couple more discoveries you need to know before you understand what's happening here. First, people found that T antigen binds a cell protein now called P53. Huge protein in cancer. Almost every human cancer has a P53 mutation. So binds SV40 T antigen. That was very interesting. Secondly, transcription of adenovirus E2 genes requires a cell protein called E2F. We talked about this before. I'll show you it again to remind you. And three, E2F, the cell protein, binds RB, retinoblastoma protein. That's another cell control protein. And P53RB and E2F were subsequently shown to be critical players in control of the cell cycle, which I will show you. Okay, here's the cell cycle. Remember, at mitosis, the cells split into two. But after mitosis, these two cells undergo cell growth, they grow DNA synthesis, more growth, and then division. All right, so this is highly regulated, and most cells in your body are not dividing. And as we mentioned before, many viruses, when they infect cells, make them divide so they have the DNA polymerase available to them. Now that's going to come into play here. Remember, a decision to go through mitosis is made by proto-oncogenes, right? If you have growth factors in the cell, in the culture medium, or in the tissue fluid, they will bind the receptor and push the cell through mitosis. So that means that there are growth factors available to let the cell divide. And all of these proteins, remember, from the growth factors, the receptors, and all the signaling down into the nucleus, they're all discovered by virtue of the fact that they're carried by transforming retroviruses. But there's a second restriction point in the cell cycle. So the proto-oncogenes may say, you can go through mitosis, but there's a second restriction point down here governed by RB protein. And if conditions are not right in the cell, you will not go through DNA synthesis and you will not do the next round of cell division. And that's regulated by RB protein. So RB protein, this was discovered in kids less than 5 years of age who have retinal tumors. And they have deleted both copies of the RB gene. You don't see these above 5 years old because the cells are no longer there in the eye. But up until 5, if you have a deletion of both genes, you will get retinal tumors. And that's because RB is a recessive oncogene. Both copies have to be lost for unrestricted cell division. If you delete both copies of RB, the cells just keep dividing and dividing. They're transformed, and they become cancers eventually. So 2 important regulatory points so far we've talked about. So here, for example, is the situation. So on the left, here's a receptor, a growth factor receptor. And when growth factors bind, a signaling transduction pathway is initiated that results in the G1S transition, so the cells can divide. So 2 signals here, the growth factor receptor-driven signal driven by oncogenes, like RAS, and a signal driven by RB. So you can see down here, RB is phosphorylated, and that lets the cells divide. How does that work? So normally RB is bound to E2F. And E2F is a transcription factor that controls proteins you need for DNA synthesis and cell division. So normally those genes are turned off in a cell. But if RB is phosphorylated, it pops off of E2F, and now E2F can turn on the genes for DNA synthesis and mitosis. So that's how RB controls the cell cycle in a normal, uninfected cell. It's by availability of E2F. Now remember, viruses need cells in S phase so they can replicate. And what they do is they antagonize RB so that they can kick cells into S phase. How does that work? So instead of RB being phosphorylated, the viruses make a T antigen that binds RB, releases it from E2F. E2F will then start transcription of all the genes you need for cell division. Again, remember, RB is bound to E2F, shutting down cell division. That's how RB is the checkpoint for cell cycle. If these are virus-infected cells, early transcription is going to produce T antigens, like large T, E1A, and E7. These will bind RB, and now E2F can transcribe all the genes needed for DNA synthesis and cell division. And that's because these viruses need the cells to be dividing in order to replicate properly. There's one more control before getting into S phase. So we've talked about the RB protein. There's another control, and that's done by P53. P53 monitors DNA synthesis. And if it sees DNA synthesis that is not scheduled, in other words, that's not scheduled by the cell, it kills the cell by apoptosis. And so if it sees unscheduled DNA or damaged DNA, so the intermediates of viral DNA synthesis look like damaged DNA, P53 can detect it. So here's a DNA break. It's detected by a protein called ATM, which activates P53. P53 then goes to promoters for genes that induce cell cycle arrest and apoptosis. So P53 is saying, something's wrong here, not a good situation. We're going to stop this cell from dividing and kill it by apoptosis. Obviously, this is not good for viruses, right? If T antigens have already antagonized RB, and now they encounter P53, what are they going to do? Well, they deal with it, of course. These are viruses. Viruses counter P53 also. And these are different T antigen ways that they do it. For example, SV40, large T, will sequester P53, so it can't work. The E6 protein of HPV causes RB to be ubiquitinated, so it's degraded. And the E1B protein of adenovirus also sequesters RB. Sorry, it also sequesters P53. And so remember, P53 is going to turn on genes that will kill the cells, so the viruses are basically antagonizing that. So the cells keep dividing and they don't go into apoptosis. So now we've overcome RB and we've overcome p53 checkpoints. Now the cell can divide. And what you may be thinking, what does it have to do with cancer, transformation of cells in cancer? We'll find out after this. Our next question. T antigens are encoded by viral genes that are essential for replication, present in tumors and transformed cells, encoded by viral genes that have been incorporated into the cell genome, are antagonists of cell cycle checkpoint proteins, all of the above. Okay, let's see how we did. All of the above is right. Most of you got that. They're essential for replication. They are present in tumors and transformed cells. They're encoded by viral genes that have been incorporated into the cell genome, antagonists of cell cycle checkpoint proteins. Okay, we're almost at solving this problem. So first, why are all viral genes gone in transformed cells by SV40, polyoma, and adenovirus? Well, but the T antigens, why are they gone? And why is transformation so rare? Well, transformation is rare because of two low probability events. First, the lethal late genes must not be expressed, right? That's our definition of semi-permissive. You get up to DNA replication and the late genes are not expressed. How does that happen? Well, one way is by infecting semi-permissive cells where the virus can't get past that DNA synthesis. So now the late genes are not expressed. Or sometimes they get deleted. The late genes are spontaneously deleted because DNA always recombines, and that can cause it as well. You lose the late genes and now you don't kill the cell. And secondly, the T antigen has to be on all the time and transmitted to every cell. And that happens by integrating it into the host DNA. So if one of the requirements here is that all these T antigen genes are integrated into the host DNA. Transformation and tumor formation, which follows transformation, are abnormal processes for these viruses. They are not needed for viral reproduction. What is needed is T antigen to turn on DNA replication. But if other events happen, like integration of those into the host cell and silencing of lethal late genes, then you have transformation. We discovered these by infecting the wrong hosts, because the right hosts for these viruses kill the cells and you never see transformation. However, we now know that even in the natural host, these events can occur. So for example, human papillomavirus causes cancers in people. And that's the normal host for HPVs. I'll get to that in a minute. So transformation is an epiphenomenon of a unique reproductive cycle. So an epiphenomenon is a secondary effect that arises from a process but doesn't influence it. So DNA viruses must start the cell cycle so they can reproduce. T antigen turns on the cell cycle and they inactivate RB, and they also inactivate p53 to block apoptosis. So if these T antigens are present and they've already inhibited these two checkpoints, then the cells divide forever, they're transformed, and they're on their way to becoming cancer cells. Okay, now we can explain HeLa cells and Henrietta Lacks. You may have read this book by Rebecca Skloot. HeLa cells. So Henrietta Lacks had a cervical tumor. In the 50s, they took a piece of it and they put it in cells in culture, and they grew forever. They're transformed. HeLa cells are present to this day in laboratories because they're transformed. The genome sequence of HeLa cells revealed human papillomavirus type 18 integrated into chromosome 18. So HeLa cells are from Henrietta Lacks. And the only intact viral genes present are E6 and E7. Nothing else is present. And so E7 binds RB and E6 degrades P53. That's why she developed cervical cancer, because randomly these two genes broke off and integrated into the host cell, made them divide forever, and eventually became a tumor. This is a very rare event in humans. Most people infected with HPVs will not develop cancers. So here's the summary of what we found today. We have learned all about the control of the cell cycle. we have found that dominant oncogenes carried by retroviruses drive cell division up the top here. The DNA tumor viruses target recessive oncogenes like RB and also P53. They target these tumor suppressor genes. And all of this is an epiphenomenon of a unique reproduction cycle. RNA tumor viruses have to integrate, and DNA viruses have to turn on the cell cycle. They're not necessary for, it's not necessary for them to transform cells, but they do have to integrate and turn on the cell cycle, and so accidentally that will lead to transformation and eventually cancer. And so it's amazing what we now understand from studying RNA and DNA tumor viruses. And what's more amazing, it all started with a chicken in 1909. All right, next time we will start to talk about how to prevent viral disease. We'll start with vaccines.